Hi, CMB students. My name is Michael Hicks. I'm an assistant professor in the Department of Physiology and Biophysics and a member of the Sue and Bill Gross Stem Cell Research Center, and I'm really excited to be able to share my research with you today. As you know, human pluripotent stem cells are able to form every lineage and cell type in the body, and I'm very interested in how human pluripotent stem cells go on to form skeletal muscle. We follow human developmental cues during every stage of development in order to form skeletal muscle progenitor cells. And these progenitor cells can go on to form skeletal myotubes in a dish or skeletal muscle fibers in vivo. And these skeletal muscle progenitor cells are marked by the marker PAX7. And PAX7 cells are highly regenerative cells, one of the most regenerative cell types in your body. They support the multinucleated muscle fibers. One unique thing about pluripotent stem cells is they can be used for personalized medicines. So you can take pluripotent derived cells from a person with a muscle disease, and you can correct that in a dish using things like CRISPR-Cas9. And then when you differentiate it back into a skeletal muscle cell, you can transplant it in and study cell therapies and how muscle regeneration works. In my lab, we use the MDX NSG mice. These are mice that model the disease Duchenne muscular dystrophy, and NSG is an immunocompromised mouse model. So we apply cardiotoxin to the skeletal muscle of the mouse, and this induces a muscle injury. And then we transplant in 1 million skeletal muscle progenitors into that muscle injury and look at how it can repair the tissue over time. And this image that you're seeing here is 30 days post-injury, and in red are the human nuclei, and green are the myofibers. And the MDX mice lack a protein called dystrophin, and so in green is that marker for dystrophin, and it's only expressed by the human cells that are now expressing it and restoring protein and therefore function to the skeletal muscle. And some of these human will go on to fuse with the mouse, and they'll form these chimeric large multinucleated myofibers, while other human cells will just fuse with themselves. They'll provide a unique opportunity to study how skeletal muscle fusion from human works in an in vivo system. This is a video of what this looks like at high magnification. And so you can see the mouse fibers here in gray. So you can see the mouse fibers here in gray, and you can see the human nuclei here in red, as well as the human nuclei that have went on to fuse and form these dystrophin-positive myofibers. And we're interested in not only are the ability of these transplanted cells to make new muscle fibers, but what happens to all these other human cells that were unable to fuse and to make muscle fibers? Some of them may retain the marker PAX7. So during my postdoc, I was involved in a really interesting project that derived a single cell transcriptomic atlas of every stage of human development, starting in week 5 in the embryonic stage, all the way through adulthood. And this is shown here in this diffusion map. And then we were able to overlay every single developmental state onto a map. And so in red, you can see here are the adult cells. These are a later stage fetal and an earlier stage fetal. And we asked the question is, what type of cells are we making from pluripotent cells? And for pluripotent cells, you can see that they align really close to week 9 and 10. And this is really interesting because when we transplanted our cells, and I showed you all these really small human fibers, we might expect that they'd behave more similarly to a fetal week 9 or 10, and less so like the adult muscle. And so we asked the question of what do these cells look like in vivo when they're transplanted versus tissues taken from different stages of fetal and adult. And so here's fetal tissue, and it's marked here by these small muscle fibers. It's a marker called spectrin. Here's the fetal tissue PAX7 cells. And as tissue develops over time, these start to condense, form a niche. And then in adulthood, these muscle fibers become very large, and the Pax7 cells are squished between the basal lamina of the myofiber and the sarcolemma of the myofiber in what we call a supportive niche. And as you can see from the cells that are derived from pluripotent cells, when you transplant them, they look much more like a fetal week 9 or 10 cells. And this enables us to have the first model of human development in an in vivo system, which is quite fascinating. And we can understand both development as well as how are the muscle fibers and stem cell niches able to support the Pax7 cell. These are really interesting questions because you can develop cell therapies that would provide a lifelong cure for people with muscle diseases. One area that my lab is really interested in pursuing and would serve as a potential PhD project are emerging technologies for single cell lineage tracing after transplantation. And we would like to adapt the approach taken from Samantha Morris' lab using a technology called cell tag. And this has a bunch of virus that have unique barcodes, and these viral libraries are then infected into cells. And now the cells have, every one of them have a unique tag. And so you could take these cells here and you can do lineage tracing. And the plan would be to take the cells and graft them into the MDX NSG muscle, and each one of the cells would now have a unique barcode. If you applied an injury, you'd be able to see whether these Pax7 cells are able to regenerate new muscle tissue, and whether the new muscle tissue, as shown by my graphic, would then be derived from a common progenitor. So are these two cells from the same parent? Are they unique cells? And are these myofibers and these cells also unique or not? So the idea would then be to isolate these cells, perform single nuclear RNA sequencing, and derive to see whether these cells are from a common parent. This would allow us to understand how self-renewal occurs and whether the niche is important in the self-renewing process. So if you manipulated any stage during here with different types of drugs or with overexpression of niche factors that allow the muscle to better interact with the muscle stem cells, you can study self-renewal in vivo and better develop cell therapies. My lab's also interested in regenerative therapies. And so we have a number of different transgenic mice. These mice here are called Pax7 Cree ZS green mice. So if you give the mouse tamoxifen, all of the Pax7 cells light up green. You can also notice that the myofibers here are also lit up in green. And that's because some of the Pax7 cells that are nearby these myofibers have fused. And when they fuse with the myofiber, they release their content, and the whole myofiber will now light up with the green fluorescent protein. So we took a 2-photon microscope and we put a mouse under anesthesia, and we could study how does regeneration occur. And so we also like to have fun with tools like Amaris, and this is what this video looks like. So you can measure all of the different muscle fibers and their different sizes and how regeneration is occurring, as well as the number of Pax7 positive cells here, and whether the Pax7 cells are binding onto these myofibers, they're being able to migrate to sites of injury. And I really believe that the 2-photon microscope, which we have here in our department, would be a fantastic tool to develop a PhD project based on. And if you're more interested in this type of work, you could check out this cell stem cell paper that looks at how the muscle progenitor cells use the architecture of dead muscle fibers to migrate and fuse to make new muscle. And we can study this with different types of drugs and different types of disease models to better understand the regeneration process. My lab's located here in the Sue and Bill Gross Stem Cell Center. It is a fantastic place. I think that it's a unique training environment full of professors that study stem cells and a number of tissues, and there's a number of great experts here. My department of physiology and biophysics is highly supportive. They're a really great group of professors, and it's a really great training environment for students. In addition to Irvine, I believe that the skeletal muscle stem cell community is among the best. They have a number of small conferences with great scientists, and they are highly supportive of trainees and have a number of training workshops. And I would encourage graduate students in my lab to pursue career development, including presenting research at science conferences. So if you're interested in learning more about my research, please reach out to me at mrhicks1@uci.edu. I look forward to hearing from you. Take care.